Good morning everyone. So myself Dr. Charan. I am working as an assistant professor in the Department of Bioinformatics, Vignan Foundation for Science, Technology and Research. So in this discussion I am going to discuss about germ theory of diseases. Before going to the concept, I want to brief about the vision mission statements of our department. So the vision of our department is to be a leading innovation center in bioinformatics, offering dynamic curriculum aimed at producing globally competent, employable, entrepreneurial and research professionals with ethical values capable of addressing challenges in multiple domains. So we have 3 mission statements. The first one, offering the state of art curriculum with skill based practices in teaching and learning to equip bioinformatics students for employment. And the second one, to provide advanced computational and laboratory resources to prepare students for higher education and research. The third one, shaping well rounded professionals with ethical values, leadership qualities and teamwork skills for lifelong learning. And also we have 3 program educational objectives. The first one, identify and analyze biological data using bioinformatics tools to design drugs through structural biology approaches. The second one, to develop predictive solutions to explore biological processes focusing on genomics, proteomics and metabolomics. The third one, demonstrate technical expertise, communication and entrepreneurship to foster professional growth and interdisciplinary leadership. So we have 2 program specific outcomes. The first one, to develop bioinformatics algorithms and tools to address key challenges in biotechnology, including genome assembly, protein structure prediction and molecular docking. The second one is to facilitate the use of databases and software tools to analyze and interpret data, providing domain expertise to support research in life sciences. So with this I want to enter into the current discussion topic, the germ theory of diseases. It has the following contents. So the first one, the introduction to the germ theory of diseases and the pre-germ theory concepts. So the pioneers of germ theory, the Louis Pasteur's contributions, Robert Koch postulates and the significance of Koch postulates and the impact on the public health, limitations and modern adaptations of germ theory of disease and summary. So this concept, we have the objective of to understand the evolution of concept of germ theory of disease and Koch postulates for identifying the relationship between the disease and disease causing agent. And the outcome, the student will be able to understand the germ theory of diseases and Koch postulates. So introduction to the germ theory of diseases. So initially, before the germ theory of diseases, people used to believe that the diseases are caused by some curse, some bad air. There are previous other concepts. So they don't know why these diseases are occurred. They don't know the causative agent. So the germ theory proposes that microorganisms are the cause of many diseases, and it replaced the earlier theories like miasma theory, which proposed that diseases are caused by bad air. This laid the foundation of modern microbiology, hygiene and infection control. Let us look into the pre-germ theory concepts. So miasma theory, it says that the disease is caused by foul smelling air. So according to this theory, so the diseases are caused by bad smell. So people that time, they are because of this theory, so till the end of the 19th century, people used to believe this and they used to follow the sanitization practices so that somehow they get benefited because they have followed all the sanitary practices and hygiene. So, because of that they are protected from the spread of disease. But next, the spontaneous generation theory. So, as per this spontaneous generation theory, it says that the life arises from the non-living matter. So, this theory was proposed von Helmont and also supported by John Needham. So, they conducted few experiments. The life arises from the non-living matter. And the next one is the humoral theory. So, it was proposed by Hippocrates, supported by Galen. So, according to this theory, the imbalance of the body fluids. So, the imbalance of the body fluids such as blood, plum, yellow and black bile are responsible for the causing of disease. Pioneers of the germ theory. At first, Girolamo Fracastoro, he suggested that invisible seeds of contagion. That means, so he proposed that disease is spread by invisible seeds. There are some infectious agents that are not visible, which are causing the disease, and they are spreading through the contact, air, and also through fomites. And Anthony van Leeuwenhoek, he first observed microorganisms under the microscope and in the pond water. So, he called them as animalcules. Later, Ignaz Semmelweis, so he advocated the hand washing. So, the hand washing will prevent the spreading of the disease. Later, John Snow, he linked the cholera to contaminated water. So, earlier people were affected from cholera, but they do not know the cause. So, finally, he given the relationship, so between the cholera and the contaminated water. So, he said that the water is the cholera is spreading through the contaminated water. So, Louis Pasteur done enormous work, so to support the theory of biogenesis. So, he disproved the spontaneous generation and he proposed the theory of biogenesis. So, by conducting swan neck flask experiment, and also he showed that fermentation is caused by microorganisms, and he also developed pasteurization is a method of sterilization process proposed till now. Now also we are currently using for the sterilization of the milk in households and also in the breweries. So, he also proposed that microbes can also cause the disease, but he did not provided the experimental evidence. So, since it was properly explained by Robert Koch. So, this is the experiment conducted by Louis Pasteur, the famous swan neck experiment. So, at first he has taken a swan neck flask. So, you can see the S shape, it has the S shaped neck. So, he took the nutrient broth and he boiled it. So, then he incubated this swan neck flask, which was boiled for few days. So, and he found that it stayed without any contamination. There is no growth in the broth. So, he again tried with one more flask, which was placed tilted, so that he found that after within hours and days he found the growth of microorganisms in the broth. It proves that the swan neck of the flask so could supply air, and also but it could trap the entry of microorganisms into the broth, so that the broth will not be contaminated. So, this experiment supports the theory of biogenesis. The life is not originated from the non-living organic matter. So, but it is actually because of the microorganisms. So finally, the confirmation of the spread of disease by microorganisms was given by Robert Koch. So he proposed 4 postulates, which was given by his mentor John Henley. So it has means he proposed 4 postulates. The first one is the suspected pathogenic organism should be present in all cases of the disease and absent from the healthy animals. This is the first postulate. According to that, the suspected pathogenic organism, whichever is the causative agent of the disease, will be present all the cases of the disease, but it is absent in the healthy animals. You can see here this is the diseased animal, this is the healthy animal. So when they have taken the blood sample and observed under the microscope, so if you compare both the samples, here only RBC are there, here along with the RBC there are certain microorganisms which are present, but which are absent in the healthy animal. Okay. Then the second postulate, the suspected organism should be grown in pure culture. So they took the sample from the diseased animal and they have cultured in a nutrient agar media. So they found the pathogenic colonies in it. So when they cultured the sample from the healthy animal, they did not found growth of any microorganism. The third postulate, the cells from the pure culture of the suspected organism should cause disease in the healthy animal. Now we have here the pure culture of the microorganism was prepared. If we inoculate this microorganism into the healthy animal, it should cause the same disease, in the healthy animal. So because of that, again disease was induced here. So the fourth postulate, the organism should be re-isolated and shown to be the same as the original. So from the this diseased animal, we should be able to prepare the pure colony. Again, we should be able to prepare the pure colony when we cultured in the nutrient agar, and it should be the same as before, and these two pure cultures should be same. Then only it says this particular disease is caused by this particular microorganism. So these are the 4 postulates for the identification of the disease causative agents. The significance of this Koch postulates are, it provided scientific proof of causation of the disease, and it was applied successfully for anthrax. He identified the causative agent of anthrax, Bacillus anthracis, and also the tuberculosis, Mycobacterium tuberculae caused the tuberculosis, and also cholera caused by Vibrio cholerae. So these Koch postulates also has fewer limitations. So the limitations are, it is not applicable to viruses, because the viruses cannot be cultured on a solid media without any host. So they are obligate parasites, they cannot be cultured outside, they need a host. And unculturable microbes, few microorganisms cannot be cultured in the nutrient agar. And for polymicrobial diseases, sometimes a disease is not caused by a single microorganism, it is caused by more than one microorganism. During that conditions, even if a single microorganism we are able to culture, so it may not if that culture is given to the healthy animal, it may not develop the disease, because the development of disease, the onset of disease is not solely dependent on one particular microorganism, it is because of the more than one microorganism. So the impact on public health. So this revolutionized the sanitation and sterilization practices, so which are very much essential in the hospitals, especially during the surgery. So where the patient body is opened, which made it more prone for the entry of airborne contamination, and also various surgical instruments are used during the surgery. So, if the microorganisms are present on the surface of surgical instruments, they may also cause some secondary infections in the patients. So, to avoid that, so as people are aware of because of the germ theory, they are aware of the diseases are caused by microorganisms, they are using the sanitization, sterilization practices, so that whatever the surgical instruments and used for the surgery are sterilized properly before using. And also it enabled vaccine development and antibiotic discovery. Now, because of this germ theory, we come to know that microorganisms are causing the disease. Now, to prevent that, scientists developed antibiotics, so which can kill the specific microorganisms, and also vaccines. So, vaccines are developed as a preventive measure, so that by using the vaccines, the host immune system will be trained before it encounters the specific pathogen. And it also introduced aseptic techniques in surgery and laboratories, and it led to the modern epidemiology and infection control methods, so that we are able to control spread of the disease, and also we can prevent the onset of any infections in the healthy people. So, the limitations and modern adaptations for the germ theory. So, asymptomatic carriers, some people may carry the microorganism, but they will not show any symptoms, because means the infectious agents may be present in animals. So, it may not show any symptoms in the animal, but it may, if it is transferred to the humans, it may cause the disease. So, because of some immunological factors, if the person's immune system is so stronger, that can counteract that particular microorganism. So, in that conditions also, it will not show any disease, and if it is transferred to any other healthy person which are susceptible, so that can cause disease. And molecular coach postulates for pathogens identified via genetics. So, during these asymptomatic carriers, how can we identify these microorganisms? Through the molecular tools. So, like RT-PCR we can do. So, through the molecular techniques, we can identify the presence of a specific microorganism that is causing the disease, and the importance of host pathogen interactions and immune responses. So, here, because of this, one more point added to the Koch postulates that the antibodies against the specific microorganism causing the disease also should be developed in the infected person, so that if the person is developing antibodies against a particular disease causing pathogen, it indicates the development of the infection or disease in that particular person. To summarize, the germ theory shifted disease understanding from mystical causes to the microbial agents. So, as earlier they do not know why the diseases happen, they are thinking some curse, some bad air, but it has provided the scientific evidence that the disease is caused by a specific pathogenic microorganism. And the contributions of Pasteur and Koch laid the foundation for medical microbiology, and it continues to influence the research, the diagnosis, and public health strategies. So, few questions that need to discuss, that is, what are the major difference between the germ theory of disease and the miasma theory. So, the miasma theory says that the disease is caused by bad air, but the germ theory provides the strong evidence that the disease is caused by a specific microorganism. The second question, the Louis
described Louis Pasteur's Swan neck flask experiment and explained how it disproved spontaneous generation. Though as per the spontaneous generation, the life arises from the non-living matter. So, these Swan neck experiments clearly proves that the microorganisms, means the life, new life arises from the pre-existing cells, because in the experiment when the sterilized broth was exposed to air, then only the growth of microorganisms was observed. If it was unexposed, then there was no growth of microorganisms within the broth. And third question, what are Koch postulates and why are important in microbiology? So, the 4 Koch postulates we have discussed. So, the Koch postulates will provide for the identification of microorganism and the disease it is causing. It establishes the relationship, the 4 postulates. And why are Koch postulates not applicable to all microorganisms such as viruses? So, because the viruses are obligate parasites, they need some host, so that we are unable to grow these viruses on a nutrient agar. And how did the germ theory of disease change medical practices and public health policies? So, because of the germ theory, they come to know that the diseases are caused by microorganisms, so that they have started using all the sanitary practices, okay, all hygienic methods, antiseptic surgical processes, so that they can stop the spread or cause of the disease through the microorganisms. These are the references, and thank you.